Hello class, welcome back. This is going to be our fourth lecture video on Unit 4, Photosynthesis. In this lecture video, I'm going to discuss a concept referred to as a photosystem, because we're going to hear a lot about photosystems upcoming when we start going into the nitty-gritty details about the light reactions, the first step of photosynthesis. So let me kind of bring you to where I'm going to be discussing. I want to bring your attention in on one of those little tiny discs here within the chloroplast, which we call thylakoids. And if we zoom in on one of the thylakoids, we're going to be looking at a lipid bilayer. And in that lipid bilayer, where my laser pointer is where I'm going to see a large grouping of pigments. Chlorophyll A, chlorophyll B, carotenoids, xanthophylls, all the different types of accessory pigments. And that's what we're going to refer to as a photosystem. So, a photosystem is going to be, like I just mentioned, a collection of pigments that are going to be found within the thylakoid membranes themselves, each and every one of them. All right. These photosystems, or these large groupings of pigments, what happens is when they get struck by light, they become excited by photons. Photons is a fancy word used to describe a beam of light energy. Think of it that way. So a beam of light energy, or photons, which cause then each pigment molecule to temporarily lose an electron. That's what I mean by excited, that I have in bold up there in blue. So over here, you can see light energy is striking. Here's my thylakoid membrane when it's zoomed out. And this big green blob right here that you see is a photosystem. And every little dot you see within it is a pigment. Chlorophyll A's, B's, etc. So zooming out, here's a large photosystem as well as what it just zoomed in on. So if this is a single chlorophyll molecule, right, and I'll drag my laser pointer over to one of these little green dots behind, that's what I'm referring to. So when light energy or photons hit it, it'll start to vibrate that chlorophyll molecule, excite it, and it will cause one of its electrons to be raised to a higher state, an excited state. In essence, you're kicking an electron out from one energy level to an outer energy level where it doesn't belong. When that electron, you can see over here in this diagram, bumps up to a higher energy level, it could then theoretically react with any other atoms or molecules that are nearby. So the excitement of one electron could end up interacting and exciting another molecule right by it. So when you look at light over here again striking a photosystem, when one gets excited and doesn't lose an electron but temporarily gets an electron excited, it'll kind of reflect through the whole complex here. And that's how you're ultimately being able to actually absorb light energy. So again, looking at this diagram here, thylakoid, zooming in on the thylakoid membrane. Here is a photosystem. All right, so it's a large protein complex made of many smaller ones because each pigment or chlorophyll A or B molecule is a protein itself. So here's a photon of light, all right, or one beam of light. It's going to excite this pigment where it has one of its electrons get raised to the higher state. That electron then, remember when it jumps to the next energy level, will interact with this pigment molecule, causing it to have one of its electrons get excited. And that keeps happening. One pigment molecule that's getting hit by a beam of light, or photon of light, will stimulate and excite another pigment, which stimulates and excites another pigment, etc., etc., till you get to an N molecule of chlorophyll. Usually it's chlorophyll A. Right? This last pigment molecule also becomes super excited, and it will lose a pair of electrons. All right? Not just get a pair of electrons excited and then they come back down. This chlorophyll molecule in the photosystem will lose a pair of electrons, and as we're going to see during the light reactions, that pair of electrons will then be donated into a protein chain that we call the electron transport chain. So as these electrons that were just lost by this photosystem begin to move through the chain, we're going to use that movement of electrons to do two things. create the hydrogen ion gradient that will be used to make ATP, as well as these two electrons will reduce NADP plus and make it become NADPH. So one more time, looking at just a different vantage point of it, this once again would be a photosystem. We have chlorophyll molecules, which are green, and I like this one too because I have some accessory pigments mixed in. So these molecules here that are red, yellow, or orange, they're going to get excited by green light, right? The wavelength of green, while chlorophylls A's and B's that are all mixed in here in green, we're going to get excited by what? The blues, the violets, and the reds. So again, once light or photons strike these pigments, this pigment complex in the thylakoid membrane, all right, what's going to happen is they get excited and one of their electrons jump to a higher state. So think of the arrow here, the point here is where that electron in this particular accessory pigment is getting excited. It will drop back down and become relaxed in a little bit, but then it would get re-excited again by more light hitting it. But that excitement of electrons will excite electrons in other pigment molecules, and you can see how they all just kind of go in one direction until eventually you get to what they call a reaction center, which is, if you look back at this other diagram here, would be this last chlorophyll molecule, chlorophyll A. And that is the chlorophyll that ultimately actually loses a pair of electrons. So those electrons are going to come back down when the pigment becomes relaxed. It has lost those electrons. Those electrons will help create the hydrogen ion gradient and eventually get to, as I just said, the final electron acceptor, NADP+, which then becomes reduced to NADPH. And we're going to use NADPH here in the stroma, right, of the chloroplast, not the stoma, which are the openings of on the leaf, right, between the guard cells. The stroma, which is where the Calvin cycle will take place, where sugar will be made later on. One last thing, that reaction center, that last chlorophyll A molecule that does lose a pair of electrons, those electrons have to be paid back. They have to be replaced. So water, remember that's needed for photosynthesis, water is what's going to replace those electrons. Water will be split. An electron from each of those two hydrogens that are in the water molecule are what replace the two that are lost. When water is split and the electrons are removed, you're left with two things. You're left with two hydrogen ions that are going to build up within the lumen, that interior portion of the thylakoids, and you will get oxygen as a waste product. And oxygen will then later diffuse out of the chloroplasts into the spongy mesophyll area before it exits into the atmosphere through one of the stoma or stomata, the openings between the guard cells.